Hello and welcome to Bio40 Pre-Class Lecture 14. Today we're going to continue talking about mutations and get into how they're repaired. We ended last time discussing spontaneous mutations. We talked about how tautomeric shifts lead to mispairing and replication. Also during replication, looping out, or slippage, can cause insertions and deletions at single base, dinucleotide, and trinucleotide repeats. We also talked about post-replication damage in the form of depurination, deamination, and oxidation. But mutations can also be the result of certain environmental factors. These factors, such as radiation and certain chemicals, are called mutagens, and we'll discuss several types. Each type has a different mode of action and leads to different types of mutations. Note that the use of the term induced is different here than when we were talking about Lamarckian evolution. This is not tall trees inducing giraffes to have mutations for longer necks. Mutagens increase the frequency of mutations, but the effects on phenotype are random. X-ray radiation can damage DNA by creating double-strand breaks. When two double-strand breaks occur in the same chromosome, we can get large-scale deletions and rearrangements, which we'll discuss later. But even one double-strand break can lead to small deletions due to imperfect repair. And low-dose X-ray radiation can increase the amount of free radicals in our cells, leading to point mutations, as we discussed last time. The other type of radiation that can cause mutations is ultraviolet, or UV. UV radiation can cause the formation of pyrimidine dimers. The carbons at positions 5 and 6 of adjacent pyrimidines become covalently linked in a cyclobutane ring. This distorts the DNA, and the normal DNA polymerase cannot read past this point. The replication fork stalls. To allow replication to finish, a different, error-prone polymerase is brought in. But this can lead to mispairing errors at and near the site of the pyrimidine dimer. Most of the other things we think about as mutagens are chemicals that either modify DNA or cause errors during replication. Proflavin is an example of an intercalating agent. Notice its planar 3-ring structure. It sort of resembles the 3 rings you see with a purine-pyrimidine pair. Due to this structure, it can intercalate, or move in between, the base pairs in our DNA. During replication, this leads to the insertion or deletion of single base pairs. We can see how this works here. If an intercalating agent is in between 2 bases on the template strand, the polymerase interprets this as an extra base, but can't tell what it is. So a random base is inserted in the new strand. If an intercalating agent inserts itself into a strand being newly synthesized, it can cause a polymerase to accidentally skip over a base, leading to a deletion. Intercalating agents essentially induce slippage. While we talked about deamination last time as a form of spontaneous mutation, there are also chemicals such as nitrous acid that can cause deamination to occur. We already addressed cytosine being converted to uracil, which causes GC to AT transitions. But deamination can also convert adenine to hypoxanthine, which pairs with cytosine. This leads to AT to GC transitions, so deaminating agents can cause transitions in both directions. There are also mutagens known as alkylating agents, which add alkyl groups to bases. For example, ethyl methane sulfonate, or EMS, can attach an ethyl group to the oxygen of guanine's carbonyl. This forms O6-ethyl guanine, which pairs with thymine. After the next round of replication, the thymine will pair with adenine, and we'll see a GC to AT transition. Other alkylating agents can affect different bases and cause other sorts of transitions. The last class of mutagens we need to discuss are base analogs. These are atypical bases that can be wrongly incorporated into the DNA because they kind of look like regular bases. For instance, 5-bromouracil, also called 5-BU or BRDU, normally behaves like thiamine and pairs with adenine. If that were the whole story, this wouldn't be a big deal. But like thiamine, 5-BU can undergo tautomeric shifts where it pairs with guanine. And unlike thiamine, these shifts happen pretty frequently with 5-BU. So if 5-BU is incorporated opposite an adenine, but then shifts and pairs with a guanine in the next round of replication, you get AT to GC transitions. But if the 5-BU DNTP is in its rarer form and incorporated opposite a guanine, and goes back to its common form to pair with adenine in the next round of replication, you get GC to AT transitions. So 5-BU and any other base analogs that function similarly can cause transitions in both directions. So we've looked at several types of mutagens, and have seen they all cause different types of mutations depending on the chemistry of their action. Here's a short summary of what we've looked at. Tautomeric shifts can occur spontaneously in normal bases and more frequently in base analogs, and they cause transitions in both the AT to GC and GC to AT directions. Slippage can also occur spontaneously, or be induced with intercalating agents, such as proflavin, leading to small insertions and deletions. Depurination is spontaneous and can lead to all types of base substitutions. Deamination can be spontaneous, or induced by chemicals such as nitrous acid, and can lead to both types of transitions. Alkylating agents, such as EMS, also modify bases, leading to transitions. Spontaneous oxidation primarily affects guanine, leading to GC to TA transversions. X-rays cause double-strand breaks, which can cause large deletions, and UV radiation causes damage through pyrimidine dimers, which halt replication and force the use of error-prone polymerases, which can cause substitutions. Because of all the different chemical activities and types of damage, any individual repair system is usually only effective against a subset of these mutagens. So organisms have many different DNA repair systems to deal with this. And the reason we need so many is because mutations are bad for you. Germline mutations can affect the health and viability of offspring, while somatic mutations can lead to cancer. One of our major life lessons for the course is maybe don't purposely put mutagens into your body. Of course, this might cause you to wonder, how do we know something is a mutagen? We need tests to detect this. And in class, we'll discuss some of the tests that do exist. But no matter how good we are at identifying mutagens, we'll never be able to avoid them completely. And there will always be spontaneous mutations to deal with. So our cells need methods of repairing DNA damage. Errors made during replication are extremely rare. We normally expect to see less than one error per billion bases copied. DNA polymerases make few errors to begin with, and many polymerases have a proofreading activity, as we previously discussed, which catches most of the errors that do occur. Outside of the cellular environment, the error rate is considerably worse. For in vitro processes like PCR, the rate is more like 1 in 10,000 to 1 in a million bases, depending on whether or not the polymerase has a proofreading function. So clearly, something else is going on in vivo to keep the mutation rate so low. Cells have many forms of post-replication repair systems, and we're going to focus in on two main classes: excision repair and mismatch repair. In class, we'll talk about how scientists were able to discover the genes involved in these processes. But for today, we'll just look at how they work. Excision repair can be divided into two types. In nucleotide excision repair, which deals with major lesions, the phosphodiester bonds are broken, and the full nucleotide, base, sugar, and phosphate, are removed. In base excision repair, which deals with more minor base modifications, the first step is simply detaching the abnormal base from the deoxyribose sugar. Let's start with nucleotide excision repair. This process fixes major distortions in the DNA, such as those caused by pyrimidine dimers. Proteins UVRA and UVRB form a complex that detects such distortions. When they do, UVRA is released and UVRC is recruited. The new complex cleaves the phosphodiester bond at two spots on the strand with the lesion. One nick is 7 nucleotides away in the 5' direction, and the other is 4 nucleotides away in the 3' direction. Then the helicase UVRD unwinds that stretch of DNA, releasing the damaged section of the strand. DNA polymerase I fills in the gap with the proper nucleotides, and DNA ligase seals the nick in the backbone. Individuals with defects in their nucleotide excision repair system have a condition called xeroderma pigmentosum. Recall that pyrimidine dimers are caused by UV radiation, so individuals that can't fix these lesions are particularly susceptible to damage from the sun. As a result, they have a much higher incidence of skin cancer and tend to develop it early in life. Inheritance is autosomal recessive, suggesting that one copy of each gene in the repair pathway is sufficient. The other type of excision repair is base excision repair. This is used to remove modified bases, like 8-oxoguanine or uracil. Enzymes called DNA glycosylases detach abnormal bases from their sugar, but do not do anything to the backbone. This leaves behind an apurinic/apyrimidinic site, thankfully abbreviated to AP site. There are many different DNA glycosylases, and each acts on a certain type of abnormal base. So there's one that recognizes and removes 8-oxo-G, another for uracil, and so on. Next, the enzyme AP endonuclease breaks the phosphodiester bond on both sides of the AP site. A DNA polymerase adds in the correct nucleotide, and a ligase seals the backbone. In E. coli, There are 2 glycosylases just for dealing with the oxidative damage that gives rise to 8-oxo-G. One removes the 8-oxo-G base directly, but the other targets adenines that are mispaired with it. After all, if the 8-oxo-G is removed from that mispair, the polymerase will replace it with a thymine and not the correct guanine. The mispaired adenine must be replaced with a cytosine before the 8-oxo-G can be correctly replaced with the normal guanine. And just replacing the A with the C means the cell can undergo another round of replication before the mistake is made permanent. 8-oxo-G is such a serious problem that there's even a third enzyme, whose job it is to hydrolyze the DNTP form of 8-oxo-G before it can be wrongly incorporated into the DNA chain. The other major process I want to cover is mismatch repair. Let's say we have a mismatch. Maybe a G was in the rare tautomeric form and mispaired with a T. Both bases are normally in DNA, so how does the cell know which one to remove and replace? Well, it's reasonable to assume that the template strand has a right base and the newly synthesized strand has the error. Now the issue is, how does the repair mechanism know which strand is old and which is new? Well, in E. coli, the enzyme DAM methylase adds a methyl group to adenines in the sequence GATC. Just after replication, the template strand will be methylated, but the newly synthesized strand won't be yet. We say the DNA is hemimethylated. The protein mut-S recognizes mismatches and recruits mut-L and mut-H. The complex moves along the DNA until it finds a hemimethylated site. Then it cleaves the phosphodiester bond of the unmethylated strand at the GATC site and at the mismatch. The helicase UVRD unwinds the DNA, and an exonuclease chews up the incorrect strand. DNA polymerase III replaces the strand, hopefully with no mismatch this time, and ligase seals the backbone. There's only a narrow window this can work in, though. Once the GATC sites on the new strand are methylated, the strands can't be differentiated. The repair system just has to pick randomly, and is wrong half the time. Humans have homologs to mut-S, L, and H, and they're definitely important for repair, as individuals with mutations in these genes are predisposed to getting various types of cancers. But it's unclear how we tell old strands from new, since we don't have GATC methylation. We have a different sort of methylation, but it doesn't seem connected to repair. Some people have suggested that the replication fork itself is key for strand differentiation, so repair would have to occur while replication is still ongoing. There are a few other ways of repairing damage that we won't discuss in as much detail. One is direct reversal of the damage. For instance, the enzyme alkyl transferase directly removes the alkyl groups that are attached to bases by alkylating agents, and photolyase fixes the cyclobutane rings in pyrimidine dimers. There's also something called SOS response, though it's really less about preventing mutations than allowing the cell to survive with DNA damage. I mentioned before that big lesions like pyrimidine dimers can stall replication forks, and this can lead to cell death. But the uncommon presence of single-stranded DNA associated with stalled replication forks leads to the recruitment of an error-prone polymerase that can move past the lesion. Such a polymerase may cause some more base substitutions, but at least the cell survives, and hopefully mismatch repair can correct any new mistakes. Double-strand breaks, which we previously said are caused by X-ray radiation, can lead to lethal chromosomal rearrangements, such as deletions, inversions, and translocations, which we'll discuss later. So we also need repair systems that can deal with these double-strand breaks. One method, called homologous recombination, relies on using the homologous chromosome, or sister chromatid if the break occurs after synthesis, as a template for repair. Another system is called non-homologous end joining. This system doesn't use a template, but simply brings the two ends together. This can be a problem if there are multiple breaks and the wrong ends are joined. Additionally, there's a resection or end trimming step that can lead to small deletions even when there's only one double-strand break. That does it for the causes of mutations and their repair. We didn't get to talk too much about those last few repair mechanisms, but there are whole upper division seminars just on this topic. Next time, we start discussing how mutations were used to teach us about the structure of genes.